Hi, I'm Dr. Joel Mortensen. I'm the Director of Microbiology at Doctors Data. I'm also a full professor of pathology and laboratory medicine at the University of Cincinnati, and I direct the Diagnostic Infectious Diseases Testing Laboratories at Cincinnati Children's Hospital. What I wanted to do today is talk a little bit about why and how we built the 360 test. We started by talking with our clients, the clinicians, and their patients. What do they need to determine the gut health and potentially disease? What are the important pieces? The focus is on key elements for the diversity, abundance, dysbiosis, and gut health, meaning inflammatory markers and the host response pieces, as well as key bacteria that are involved in the microbiome, as well as known pathogens, known pathogenic bacteria, pathogenic fungi, and pathogenic parasites. Because there isn't just one test that answers all these questions. What we needed to build was 5 pillars, and we'll talk about those in just a second. Together, these important markers help us diagnose microbiome imbalance and gastrointestinal disease to focus on both the host and the microbiome, both sides of the equation in health and disease. Developing the 360 profile, we developed the 5 pillars. We looked at molecular testing of the key bacterial groups, phyla, order, family, genus, and species of important organisms in the microbiome to help us tell the difference between normal biotic or healthy microbiome and dysbiotic or an abnormal microbiome. We also then looked at the key focused PCR-based assays for pathogens, including then culture to look at our ability to isolate organisms of particular interest and to give us the ability to do susceptibility testing, be it prescriptive antimicrobials or botanical agents, parasitology, and again, molecular parasitology and conventional parasitology, because there aren't just 3 or 4 parasites. There's actually a host of parasites, and we can identify at least 50 different parasites that may be important in the health and the well-being of an individual patient. And then chemistry. And the chemistries we selected were so that we could understand the actual host response, the gut response for inflammation, for fatty acids, for lots of other components that are key to understand the complexities of that microbiome and its interaction with the host. The molecular microbiome diversity and abundance part of the 360 is actually a CE marked IVDR test developed to specifically look at dysbiosis or abnormal microbiome and then the healthy microbiome to differentiate those populations. So it focuses on key organisms and groups of organisms. As I said before, more than 300 phyla, class, order, family, genus, and species of groups of organisms that we know are associated. And so we really started with hundreds and hundreds of primer sets, PCR primer sets, and focused them down until we got to exactly those groups that differentiate a normal population from a dysbiotic population. After that, we came out of the laboratory and did more than 50 clinical studies, both in Europe, United States, and Australia, to look at this large number of organisms in important populations with more than 5,000 patients and 50 clinical studies to help us understand the difference in a true clinical setting, not just a research laboratory-based test. The focused PCR assay for pathogens is also a key component, a key pillar in the 360 assay. This is an FDA-cleared assay specifically developed to be exquisitely sensitive and specific for known bacterial, viral, and parasitic pathogens, and allows it to complement the standard dysbiosis concept we were talking about from things that truly cause disease and are appropriate for antimicrobial therapy. Culture is another critical pillar in the 360 assay. And this is a conventional methodology, but it's the key to allowing us to isolate the pathogens, if we need to, for susceptibility testing. But also, we have a unique assay for testing both prescriptive and botanical agents for the appropriate use in the correct clinical setting, where we might want to actually try to affect the microbiome or directly treat an organism with an agent. The culture for us looks for a significant number of different organisms that can be involved in both pathogenic processes as well as the dysbiosis. To do that, we use a state-of-the-art mass spec time of flight with a database of more than 5,000 organisms, so we can really focus down where we need to on particular groups, species, or even strains of organisms. And then again, the culture allows us to do susceptibility testing if needed. The fourth pillar is parasitology. And there's two important aspects of parasitology. There's known pathogens, and we again use the PCR, FDA-cleared PCR assay for known parasites that cause disease. But we go beyond that. We do conventional parasitology, where we can identify more than 50 parasites that may be present in the stool sample. It varies around the country, it varies around the world, what might be there. And some of these organisms are traditionally considered commensals. But in fact, they may be involved in dysbiosis and may be something that we need to consider and look at as a cause or a player in dysbiosis. So simply limiting to a short list of PCR-compatible organisms that we know cause disease, we have the ability to go far beyond that. We're one of the largest parasitology laboratories, actually in the world, doing parasites that go beyond what almost every hospital can do in the United States because of our expertise and the personnel that we have and the experience at Doctors Data. Our focus on stool chemistry is our last and an important pillar in the 360 because it measures directly what's going on in the host gastrointestinal tract, not making assumptions about a bacterial group that may affect something, but rather what's actually going on. So we're looking at factors for absorption, digestion, and inflammation. So for example, short-chain fatty acids may well be important in the patient, or secretory IgA, or lactoferrin, or lysozymes, or others. So it's these 5 pillars that support the 360 today and provide what I think is the best ability to evaluate abundance, diversity, and dysbiosis. So Helicobacter pylori may be an important question, and it depends on the patient. Do you suspect that a patient has Helicobacter pylori? Doctors Data offers a state-of-the-art test that can detect Helicobacter pylori. Rather than search in all patients for Helicobacter pylori, where depending on the population, the incidence may be either very low or non-existent, or in fact much higher. People ask me sometimes about why don't we do pathogenic markers of Helicobacter pylori. We have the capacity for PCR detection of all kinds of various factors that may be produced or not produced. But the clinical relevance of all those is simply in question. The detection of Helicobacter pylori with clinical symptoms indicates that one needs to address Helicobacter pylori. Simply detecting that it's there or that there's a pathogenic factor there doesn't indicate a need for treatment, but simply is a detection of the DNA or the potential. So what's the role in shotgun metagenomics or next-gen sequencing in microbiome? It's a wonderful research technology. In fact, I have experience with all of them. We use them at Cincinnati Children's Hospital to evaluate a variety of fascinating conditions about the creation changes in microbiome depending on a variety of clinical conditions and what's going on with our patients. But that doesn't make it a clinically relevant assay. So in fact, I think it's a wonderful tool for understanding in a broad research sense, but it simply isn't a test methodology that yet offers us clinically relevant information for comparing normobiotic and dysbiotic and our ability to affect those when we make clinical decisions and changes in therapy. The massive data sets that are generated by next-gen or shotgun metagenomics are an amazing collection. We may be able to identify literally thousands of organisms present and look at their relative quantities in the sample. But we don't yet understand how that affects diversity, abundance, and dysbiosis. Another aspect of next-gen sequencing is the ability to detect antimicrobial resistance markers. And again, it's very interesting to develop tests to look at someone's resistome, is what we call those, and again generates thousands of data points. And in fact, we find that everyone that we test, with some limited populations being a little bit different, that we all are carrying antimicrobial resistance genes associated with all kinds of different bacteria. Soil bacteria, bacteria in the water, bacteria in our microbiome. But again, the potential, the genetic material there, and the expression are completely different concepts. And when you do shotgun metagenomics for resistance markers, you have no idea who they're associated with. So they're simply floating out there as genetic tests, may have nothing to do with the pathogen or the dysbiotic organisms. They're simply present as part of the genome of the milieu of the soup that we call stool. So we don't yet know what to do with that kinds of information. And in fact, for most of us, there'd be almost no single antimicrobial agent that would be active against anything we're looking at. And yet we isolate an organism, we do susceptibility testing, and we can cure someone from Salmonella or Shigella or Campylobacter. So that the resistome parts, I think, are going to lead to a better understanding of the movement of resistance genes in a population, but aren't yet relevant to an individual patient, their abundance, diversity, and dysbiosis.